So I'm Bill Copeland, the chief of the Genome Integrity and Structural Biology, where Sam had his laboratory for many years. I'm going to introduce Rick Wojcik. He's our director of the Institute, and he's going to say a few words about Sam and his legacy. Rick?
So let me just take a few minutes here and talk about some of the remarkable things that Sam did during his tenure here at NIEHS. So let's start off with the first slide. So it all began back in May 8th of 1996. Remember the 1900s, some of you? So it was in fact, at 12 p.m. Eastern Daylight Time, the world was alerted to the fact that there's an internationally known scientist named to the NIEHS Deputy Director. And I think the quote here from Dr. Houten, who recruited Sam to NIEHS, speaks accurately. So this is all about, with the appointment of Dr. Wilson, the Institute is gaining an outstanding researcher and science administrator. So that really says it all. So let's go to the next slide. So when Sam got here in 1996, he was the Deputy Director of NIEHS, and he brought quite a number of different scientific initiatives. One that I think was really important and has lasted throughout the course of the last several decades is this increasing recognition that it's not just about the things in the environment that you have to study, but we all react to the environment in different ways based on our genetic makeup. So he brought this whole concept of the gene by environment interactions. And I know that that was an area that was a very high priority for Dr. Houten as well. So it's something we need to figure out. We haven't quite solved it yet, but it's something that's really important, and we can really look back at some of the remarkable contributions that Sam brought to NIEHS. Now, additionally, he was also involved in the creation of the DERT, a program analysis branch, which gives us tangible data to know how are we spending the money that we have as part of our budget here at NIEHS as it relates primarily to funding grants in the extramural community. He also brought the vision of toxicogenomics and was able to establish the Toxicogenomics Research Consortium. It's really kind of in the context of that gene-environment interactions. We're looking not just at toxic chemicals, but also looking at the genomic aspects of this. He also was involved in initiating the NIEHS Environmental Genome Project and the associated Comparative Mouse Genomics Centers Consortium. So actually, this was before I was at NIEHS, well before I was at NIEHS, but I can tell you this was a very, very powerful vision to bring forward these ideas. And I got to tell you, I think NIEHS was right at the cutting edge of these things, and I think was way ahead of some of the other institutes of the NIH on these different concepts, especially as it relates to using the mouse as a model organism to study the effect of environmental contaminants. He was also involved in the creation of the Parkinson's Disease Consortium, and we're increasingly recognizing that there are things in the environment that predispose to the development of Parkinson's disease. So it's really Sam's legacy that starting this and bringing that greater awareness of the importance of looking at the environment. It's not just your genes. The genes play a role in this, but it's the things in the environment, it's gene by environment effects that are major contributors. And he also brought a global focus to the work at NIEHS. Next slide. In fact, when there was an environmental factor article, and he brought a global focus because he recognized that NIEHS's funding portfolio and interest have always spanned national borders. So it's not just about the things that happen in North Carolina or the United States, but we have to be paying attention to things that are happening across the globe. And that's especially true these days with some of the health effects associated with climate change. It doesn't stop at the borders in the United States. So Sam brought this powerful vision to bear on really looking broadly across the globe and looking broadly around how we develop these global collaborations. Next slide. And one very important function of a deputy director is to step in when the Institute needs an acting director of the Institute. So Sam did that very capably in 2007. So he was the acting NIEHS and NTP director from 2007 to 2009. And in addition to his remarkable scientific contributions, it was also really noteworthy what he set the tone as being approachable. In fact, when I first got here in January of 2011, Sam being this very famous scientist, it was just amazing how it was so easy to come into his office and how easy it was to talk to him and how much he really cared about me and cared about his colleagues. He was really known, call me Sam. I think that really set the tone. It's not about having this division between the administration and the rest of the Institute. It's really all of us working together collaboratively. If you're at NIEHS, you're important because you're contributing to the mission of the work that we need to be doing. So he's really known for his candor and his accessibility. And during his tenure, he really underscored the support for the intramural basic research program here. We do some pretty remarkable work. And I think we have one of the strongest intramural research programs across the NIH. And he also strengthened ties with the extramural community. So it's not just working in the intramural program, but also collaboratively with key investigators in the extramural community. He also increased our commitment to the journal Environmental Health Perspectives. And actually, when I was deputy director, that reported into me. If you haven't actually looked at its impact factor in recent days, you might want to take a look at it because it's really gone up. Environmental Health Perspectives is clearly one of the premier journals in environmental health sciences, and really goes back to the support that Sam brought to that journal, especially at a time when maybe there were others in administration weren't feeling quite so positive. So it's really a tribute to his strength of bringing support to the journal. And probably really important is that he reaffirmed the institutional support. It started with Dr. Houten, but it's really important to pay attention to environmental justice and community-based participatory research. So it's something that's still an important fabric of the things that we do here. And so if you're not aware of this, the Secretary of Health and Human Services recently declared that environmental justice, and this is coming from the White House, that environmental justice has to be a major focus for the US government. So we have to pay attention to some of those underserved communities where major health disparities associated with the types of environmental exposures that really fall under this moniker of environmental justice. Next slide. And he was very supportive with his gene by environment effect. He also recognized that there's really important to pay attention to what we call the epigenetic effects. So it's not just the genetic, the changes in your DNA code, but the chemical modifications to the DNA that can affect how your genes actually work. So it was really at one of our council meetings in 2008 that Sam presented on this new, very visionary program, the Trans-NIH Roadmap Epigenetics Program. It's really about bringing the genes and gene variants together with environmental exposures, and then really looking at the epigenetic signatures. The epigenetic regulation of gene expression is an emerging frontier in understanding health and human disease. Boy, that's an understatement. I mean, it's a major focus of the way that we think about how the environment is affecting how your genes are working that can ultimately give rise to adverse health effects. Let's go to the next slide. And of course, Sam was not just an administrator, he was really just an outstanding investigator, really setting the standard in DNA repair. So as we've just heard, his primary appointment was in the Laboratory of Genome Integrity and Structural Biology. He had a secondary appointment in ESCBL, the Epigenetics and Stem Cell Biology Laboratory. It's this bullet point, Sam's research was very productive. Boy, that's an understatement. I guess we're gonna hear a lot more about some of the remarkably productive things that Sam did. He really set the standard. The data shows that it was a publication per month. So that's a lot of science. And he just made major strides in understanding both the environment's role in genetic damage and basic excision repair. So here's someone who's absolutely on the cutting edge of the field of DNA repair. Next slide. And as a consequence of these remarkable accomplishments as both an administrator and a scientist, I mean, this is just a short list of some of the remarkable things. Scientist of the Year Award in 2003, Mentor of the Year Award in 2014. And that's the other thing I want to point out here. He's approachable, thoughtful, smart, but he is just a remarkable example of a mentor, someone who cares about others, actually, whether it was me as the new deputy director or trainees in his laboratory or trainees across the intramural program here. I mean, Sam really cared, and he rightfully was deserving of the Mentor of the Year award in 2014. He also received the National Institutes of Health Directors Award, which if you don't know that one, it's the highest honor an NIH researcher can receive at the NIH. So he got that in 2015. And a year later, in 2016, he was named Champion of Environmental Health Research. So this is just a short list. You can take a look at his CV, and it just goes on, page after page of the remarkable accomplishments of this wonderful scientist. Next slide. And I think I'm just going to end up here by really summing it up. It's just an incredible friend and a role model for me and for the rest of us here at NIEHS, at the NIH, and across the biomedical research community. I think Sam was an example of how you can be a top-notch, cutting-edge scientist, and you can be a warm and engaging colleague at the same time. That's not true for a lot of scientists who are at the cutting edge. But Sam, we miss you, but your memory stays with us. Okay, Bill, I think I'm going to turn the podium back over to you. I think that was the last, yep, thank you. So thank you for the opportunity to make a few remarks.
Thank you, Rick. That was a really nice summary, a brief summary of Sam's accomplishments. So I want to first thank everybody for being here, and I want to acknowledge Bob Petrovich over there, who took the lion's share of organizing this event today. So a short round of hand of applause for Bob and for talking about it. I also want to mention we did want to have it last year in August, and we picked August, of course, because this is Sam's birthday month. But as you know, last year the pandemic was still raging on, so we're happy to be here today. So it is my distinct pleasure to introduce today's talk speaker for this inaugural Sam Wilson Memorial Lecture. And in picking that person, we wanted to meet several criteria. We definitely wanted someone that shares the enthusiasm and the spirit of research and the rigor that Sam portrayed in his research. Secondly, we of course wanted somebody who worked in DNA repair. That was sort of a given. And thirdly, we wanted someone that had been influenced significantly by Sam's leadership and mentorship, and in fact followed in some of the footsteps that Sam laid out. And with that, it is my distinct pleasure to introduce Dr. Ben Van Houten, who will talk today, and a little bit of brief history about Ben. Ben received his bachelor's from the Clarion State College. After that, he obtained his PhD from University of Tennessee at Oak Ridge. From there, he accepted a postdoctoral fellowship here in the Triangle at the University of North Carolina in Chapel Hill, working for a very humble scientist that, as we know, Dr. Aziz Sankar, who, as you know, shared the 2015 Nobel Prize in Chemistry for his efforts on DNA repair. Specifically, as you know, Aziz helped discover and characterize nucleotide excision repair. And I like to think that some of Ben's work during his postdoctoral years, which was significant, helped contribute to that award. So after that really successful postdoc, Ben accepted a faculty position at the University of Vermont and worked there for several years until in 1994, he was recruited by Sam to the Seeley Center at the University of Texas Medical Branch. And if you don't know, Sam was actually the founding director of the Seeley Center at UTMB. So Ben joined that in 1994. 2 years later, we know Sam transitioned to the NIEHS as the deputy director. But in 1999, Sam helped recruit Ben to the NIEHS. And at the NIEHS, Ben actually wore two hats. He was a branch chief in the Division of Extramural Research and Training, helping to give out grants. And he was a senior investigator in the Laboratory of Molecular Genetics, running his own group on DNA repair. And as many of you know, the LMG, or Laboratory Molecular Genetics, was a precursor to the now Genome Integrity and Structural Biology Laboratory. So Ben worked here for about just under 10 years before a really incredible position opened up at the University of Pittsburgh to help co-direct the Hellman Center for the Excellence of Science. And Ben has been there ever since. Ben took that position and has flourished even further doing his work on also aspects of DNA repair, crosstalk between nucleotide excision repair and basic excision repair. He's received many awards, several numerous NIH merit awards, also the Medal of Slovak Academy of Science Award, the Miro Aragon Excellence in Science Leadership. So with that, I'd just like to turn the podium over to Ben, and we'll look forward to your talk today. And thank you for accepting this invitation.
Well, thanks, Bill, for that incredibly generous introduction. I want to extend Rick's welcome to all of you. It's so nice to see Dorothea and her family here in the front row. So it's an incredible honor and privilege to really give this inaugural lecture to celebrate Sam's scientific legacy. And I have to say, I approached this with more than a little trepidation. I chose this title for several reasons. Soon, as you'll see, this is the title of a special issue to honor Sam. But we also, in my lab, used light to interrogate fluorescent molecules. We used light to image in cells, and we used light to actually produce DNA damage. And I'll talk about these things in a moment. So what you're seeing on this screenshot is from an animation that I'll show in the second half of my talk. Let's see if I can get this to wake up. There we go. What you're looking at is a piece of DNA suspended between two beads. And this is a nucleosome between these pieces of DNA. And this is PARP, enzyme that Sam did a lot of work on, actually binding to a nick in a nucleosome. And we've been able to visualize this at the single molecule level. So before I present my scientific talk, I wanted to spend a few slides and talk about Sam. I had the privilege of serving at SAM during Sam's tenure here as deputy director and acting director. And if you're not familiar with NIH, they use a lot of acronyms. And SAM actually stands for three important scientist, administrator, and mentor. And I want to talk about each of these in turn. So, as you heard, the COVID epidemic really affected a lot of things. And as you know, Sam was the editor-in-chief of DNA Repair, the journal that you see here. And when Bill Beard and Brett Freudenthal and I learned of Sam's illness, we wanted to organize a special symposium to honor him. And just about at that time, COVID broke out. And we were like, oh, what are we going to do? Well, an amazing editor for Elsevier, Helene Hodak, and I decided, well, let's have all the speakers contribute a special graphical review, and then we'll surprise Sam with a special issue. So she worked really hard, and she was able to deliver 10 copies to Sam right at 12 noon. Here you can see Sam was expecting to Zoom with me about a scientific problem that I was having. But when he opened up his Zoom link, he saw everybody, and look at him smiling. So it turned out wonderfully. And Sam amazed all of us by going around to each person on the call and extemporaneously saying something very special about their work to each of us. And it was as if he had written it out and rehearsed it. He's just an amazing man. So when he got to Amy and Brett, he opened the article that they wrote, and it's hard to see, but this is the history of DNA polymerase beta, a very important protein that Sam spent most of his career studying. And this is a timeline of all the discoveries that Sam made on this important protein. And he looked at this and when he got to Brett and Amy, he goes, yeah, this all looks correct. You guys did a good job. So it was great. So I encourage you to read this special issue and especially this wonderful review by Amy and Brett. My graphical review of the special issue was all about Sam. And as you know, Sam loved graphs. And he would really love this graph because basically what you're looking at here is the number of publications over his entire career. So he averaged 8 publications a year for 55 years. Amazing. And what I want you to see is, you can see that his highest productivity is when he was a deputy director and acting director. So it's just a testament to his tremendous energy and drive. During the toxicogenomics years, Sam and I loved to view data as interactomes with nodes and edges. And here you can see key words that I lifted out of all of his papers, and they're linked by similarity. And I've then color-coded it over time. And you can see that in the early 1990s, HIV reverse transcriptase, hardcore biochemistry, and over time he moved to DNA polymerase beta, looking at the structure and the dynamics with Bill Beard. Then he reconstituted base excision repair with the help of Rajan Prasad. And finally, he moved into mammalian cell culture with Julie Houten. And I think this is just an amazing body of work. So I used 430 publications. He had 821 co-authors. 73 of these are on this slide. And it's broken down into different groups of research interest. It's just an amazing contribution to the field. So when Sam passed, Phil Hanwalt and Leona Sampson and I had the privilege to write an article for DNA Repair about Sam's scientific legacy. And Leona wrote, who's sitting here in the audience, I also tried to emulate this slow and precise way of taking an audience through complex data. And when Sam said, now I'm going to take you through these details on this figure by figure, step by step, it was clear that what he really meant was, now sit up and pay attention. So he was in all seriousness, and a lot of times he would be invited to meetings and he would always go, but he was so busy that he wouldn't attend things that the organizers had done for the group. But here you can see that, in fact, he was at a meeting in Italy that was organized by Jenny Dogliotti, and they went to a prosciutto factory, and you can see him hamming it up with Jenny and Leona. And so I got to know Sam when I was in Galveston at UTMB, and this was a dedication ceremony for the Seeley Center for Molecular Science. Stephen Lloyd and Phil Hanwalt, and that guy you probably can't recognize, but that's a younger me. So some of the Samisms that he would use, he had this knack of certain phrases that would resonate with people as he was leading them. Circling the wagons, I love that one. And what he meant by that was you really need to support your peers through helping them with their projects and promoting their science to others. He always talked about disciplined science and laser focus. Stay on task, get things done, and don't deviate. Good ideas are easy and implementation is hard. And I'll have a special slide about that. Problem definition, it's a term that I use in my lab all the time, and I'll explain that. And then raising the bar. So good ideas are easy, implementation is hard. Sam taught me that coming up with a good idea was relatively easy, but it's the implementation plan that really matters much, much more. He taught me that when you're developing an idea, one really needs to get buy-in from everybody involved. And in terms of NIH, from the potential grantees to the program administrators who write the RFA, to the review team who come up with the criteria to review the applications. I used to call Ken and Sam the dynamic duo. Not only did they do amazing things together, they were, as you heard, gracious, kind, and outgoing. And as you can see, they were incredibly well dressed. So Sam espoused that to be successful in science, one must select the most compelling research question to attack. And then one must acquire the best set of hands to develop and validate tools to help answer that question. Well, this is easy to say, but incredibly difficult to do. And no one epitomized that better than Sam himself. He always seemed to pick the most important scientific problem and muster the human and technological resources to solve that problem. Raising the bar. He had this amazing ability to start out a sentence and he would give you a compliment at the beginning of the sentence, but by the time he ended the sentence, he had raised the bar on his expectations for you in both an inspirational and compassionate way that empowered you to want to do great things. I remember in the old days, you would get your scores in this teletype and you had to rip it open. I was in Galveston and I was worried I was running out of money, and I opened it up and Sam happened to be walking down the hall, and I said, I got a good score. My third grant, I'm so excited. And he goes, that's great, Ben, but I know you. You're going to need 5 grants to cover all the work that you want to do. It's like, eh. So it's in this spirit. Sorry, I dedicate my talk today to Sam. I was given a very kind opportunity from Dorothea to meet with Sam just 5 days before he passed. And he said a lot of things, but one thing he did is encourage me to go all in with my single molecule work, the topic of my lecture today. So I'm going to tell 4 short stories. I'm going to tell you about a protein that was kind of in the backwaters of nucleotide excision repair, a process that we remove bulky lesions. And I'll tell you that it actually sits at the confluence between a pathway that Sam studied, base excision repair and nucleotide excision repair, and it actually stimulates many repair proteins in base excision repair. I'll tell you then about a new single molecule approach that we've affectionately named SMADNI, and you'll see why in a second. We used this approach to study 2 of Sam's favorite proteins, PARP1 and ligase XRCC1 complex. And we've been actually able to watch single molecules interrogate NIEHS on nucleosomes with this new approach, SMADNI. Then you heard about epigenetics. Well, thymine DNA glycosylase is involved in oxidative demethylation of 5-methyl-C. And I'm going to show you some unpublished data from Brittany on this amazing enzyme and how it gets turned over by UVDDB and actually interacts with DNA. So genome stability has been a wonderful supporter, been supported by NIEHS, both in the intramural and extramural program. And some of the fundamental problems in genome stability that I've worked on are, what are the endogenous and exogenous environmental factors that cause DNA damage? What are the pathways that remove them? What are the consequences of defects in DNA repair? What are the kinetics of the protein-protein and protein-DNA interactions to remove these lesions? And what are the binding motifs that proteins use to embrace the DNA? One of the fundamental questions that I've been going after my entire career is how to repair protein C lesions. And how do repair enzymes tell damaged DNA from non-damaged DNA? And how do they sort through this sea of non-damaged DNA in the nucleus to find those rare needle in the haystacks? So these last 3 points is what I'll talk about. So I have to give a little bit of background on this protein that I've been working on for the last 10 years called UVDDB. It stands for UV damaged DNA binding protein, and it interacts strongly with UV photoproducts. On a beautiful day like today, if you go outside, your skin will get UV photoproducts. This is a cyclobutane pyrimidine dimer. And it's the first protein that senses that lesion in the context of chromatin. Oddly, though, it also recognizes abasic sites, and I'll explain why that's interesting in a second. So if you get a mutation in this red protein, DDB2, it can lead to a syndrome called xeroderma pigmentosum. These people have the inability to remove these photoproducts, and if they go out in the sun, they will get skin cancer. It's just a matter of time, it's not if. Interestingly, and I'll show you a little data, a knockout of DDB2 dies prematurely to spontaneous tumors. And that's really odd because there's no UV light getting inside of them. How does that happen? And low expression of DDB2 is a poor prognostic indicator for a number of cancers. Why is that? So structurally, this is a beautiful structure that you're seeing here. So DDB2 is the damage recognition subunit. DDB1 is a scaffold that connects to colon 4A and RBX. These are proteins that are called ubiquitin ligases. Their job is to go in and modify the nucleosome and open it up to allow the other repair factors to come in. So the working model is that UVDDB sees the lesion in the context of chromatin, it ubiquitinates H2A, and it destabilizes the nucleosome, allowing the next repair protein, XPC, to come in and say, here's the lesion, guys, we need to fix this. If it sits too long, it gets removed by VCP P97. It's actually sad. Here's the first responder, but it actually auto-ubiquinates itself and destroys itself because that's the price you pay for really good recognition. So I told you, Stu Lin had shown that HETs and the knockouts actually die prematurely from spontaneous tumors. Well, that doesn't make sense if that's its only role. And if you express low levels of DDB2 in many tumors, this is just showing endometrial and cervical cancer, it's a poor prognostic indicator. So why would that be? So we started to question, does it have a more general role in the removal of other types of DNA damage, such as oxidative or alkylation damage? And is UVDDB the first responder for base damage? So I'm going to summarize 4 years of amazing work by a research associate, Sunbak Jang, in just one cartoon. What you're seeing in this cartoon is base excision repair, what Sam dedicated a large part of his career to. And you can see that this is adeoxoguanine. It's about 1 in a million bases in our cell. It's there all the time. It's recognized by glycosylase, OGG1. After removal of the oxidized base, APE comes in, nicks the DNA. That activates poly-ADP ribose polymerase, which paralates itself, which is a signal for the arrival of DNA polymerase beta and XRCC1. And then XRCC1 is to recruit ligase. And then ligase seals the nick. So Sam, using purified proteins, was able to reconstitute this entire system. What our group was able to show is that OGG1 actually doesn't turn over very well. People thought it was APE that would help turn it over, but we found it was UVDDB. In fact, it binds tightly to abasic sites. It has affinity for adeoxoguanine GC pairs as well as GA pairs. It stimulates OGG1 threefold, APE eightfold, and in fact, in a wonderful collaboration with Sam and Rashad, it stimulated DNA polymerase beta gap filling 30-fold, which was really astounding. In wonderful collaboration with Sheila David, who's probably the only person on the planet that can purify MUT-YH, we were able to show, and thankfully a reviewer asked for this, well, does it work on adeoxoguanine across from A? Adeoxoguanine is really bad because when the polymerase sees it, it doesn't put in a C, it actually puts in an A. And there's enzyme, MUT-YH, is dedicated to saying, oh, that A shouldn't be there, I'm gonna take it out and give replication another chance. Well, the weird thing is MUT-YH, after it removes the A across from the G, just sits there, and APE cannot remove it. And a wonderful postdoc who had just joined the lab, Matt Shai, showed that MUT-YH gets stimulated by UVDDB fivefold, and in fact, UVDDB can push off MUT-YH, and was a really important finding. So I had an amazing graduate student in the lab, Namrata Kumar, and I gave her a really hard task, and she took it on and did an amazing job. And her hypothesis was, does UVDDB bind strongly to, since it binds strongly to abasic sites, intermediates, can it modify the chromatin architecture? So does UVDDB recognize adeoxoguanine in the context of a chromatin in a nucleosome and help facilitate repair? Sarah Delaney at Brown had shown that OGG1 cannot find adeoxoguanine very efficiently when it's in a nucleosome. So her hypothesis was UVDDB is important for pushing the chromatin aside and allowing access by OGG1. And her approach was to use cell biology. So the very first experiment that most people do is let's look for the accumulation of 8-oxoguanine. And in this experiment, she did a wonderful knockdown of DDB2. The positive control is if you knock down OGG1, you should see an accumulation of 8-oxoguanine. There's really good antibodies from Tevagen that bind 8-oxoguanine. And you can see the positive control is nice and green. But excitingly, when we knock down DDB2, the nuclei turn green as well as compared to the control. So that said, the steady state levels of 8-oxoguanine in untreated cells actually creeped up over time if DDB2 wasn't there. And that's just quantified here. Well, a reviewer said, well, what about cell death? And actually it was Rick Wood at a meeting that brought it up. And if you treat cells with potassium bromide, which oxidizes DNA, producing primarily 8-oxoguanine, if you knock down DDB2, they're more sensitive to killing by potassium bromide. The reviewer then asked us to prove it. And so we used an assay that I can't explain today because of time, but basically if you knock down DDB2, repair of 8-oxoguanine is slower. And that was a really important finding, and Namrata worked very hard to get these data. Well, wouldn't it be cool if we could put 8-oxoguanine in a specific place and ask, does DDB2 go there in response to 8-oxoguanine? In an amazing collaboration between Marcel Bruchet at Carnegie Mellon University and Patty Opresco, they developed a system to make a singlet oxygen bomb and then deliver that bomb to the telomere. And so Marcel was an amazing chemist, and he found this protein called a fluorogen activating peptide that would bind to a dye called malachite green. Now, malachite green is flexible and doesn't fluoresce, but when it's wedged between this dimer, it can fluoresce. But he's a chemist, so he put 2 halogens on it, and in the excited state, it actually produces singlet oxygen. And if you bring singlet oxygen close to DNA, you'll get 8-oxoguanine and almost only 8-oxoguanine. So Patty got the great idea, let's fuse it to a protein that binds to telomeres called TRF1. And so the idea is that TRF1 drags this singlet oxygen bomb to the telomeres. You shine light in the presence of 660 light and dye, you produce singlet oxygen that then makes 8-oxoguanine at the telomeres. So now we have a way to say, who gets there first, UVDDB or OGG1? And so the first experiment, you can see robust recruitment of DDB2 to the telomere only after dye plus light. So this is exciting. The M-tray DDB2 is going just to the telomeres and only with dye and light. So we're putting 8-oxoguanine in, it's going there. Who gets there first? Well, immediately after dye and light treatment, 5 minutes, UVDDB is there, and then OGG1 shows up about 30 minutes later. And so that's quantified here in this immunofluorescence imaging. She also found that the second repair protein, XPC, shows up. Well, that was a surprise. And then another repair protein, XPA, shows up with the same kinetics as OGG1. So this is the fundamental question that she asked, is DDB2 required for the rapid and efficient recruitment of OGG1? So here you can see at 30 minutes is the peak recruitment. If we knock down DDB2, do we still see OGG1? And you can see there's a 3 to 4-fold reduction in OGG1 recruitment to the telomeres. And so this really said, wow, DDB2 has to go there to say, here's the lesion, and OGG1 goes there. Well, that wasn't enough for Namrata. She noticed when she was imaging that, she goes, Ben, the telomeres look actually bigger. And I said, what do you mean they look bigger? So in a wonderful collaboration with Simon Watkins at the Imaging Center of Biological Imaging, and with Ryan Barnes, you can see that the telomeres actually get bigger after the dye, but in a DDB2 knockdown, they don't get bigger. And so what we think DDB2 is doing is decompacting the chromatin and allowing access by OGG1. So this is a working model. DDB2 lands, and I didn't have time to show you. We think DDB2 is necessary and sufficient, doesn't need DDB1. It opens up the chromatin, allows access by OGG1. We think that helps recruit XPC, which also helps turn over OGG1. We're not sure if it gets destroyed after this event. And I don't have time to talk about this, but if OGG1 works at a nick and makes a nick by APE, we found that XPA actually gets recruited to help do transcription coupled repair. And that was work that Graciela Spivak with Phil Hannawalt was able to show several years ago. So this raises the big question, one glycosylase, does it stimulate other glycosylases? Well, mammalian cells have 11 glycosylases, and I told you about MUT-YH and OGG1, and we found evidence we haven't published on uracil glycosylase, methylpurine glycosylase in a wonderful collaboration with Barbara Van Loon that Leona helped set up for me. We were able to show that it actually UVDDB co-localizes with OGG at sites of MMS damage where OGG would remove it. And SMUG, single-strand specific monofunctional uracil DNA glycosylase, we just recently published a paper by Priya Raja that showed that after you treat cells with hydroxymethyl DU, you can see co-localization of DDB2 and SMUG, and DDB2 stimulates the turnover of SMUG. What about thymine DNA glycosylase? And I'll talk about it at the very end. So there are three questions that I want to answer in the last part of the talk. How do repair proteins find rare damage in a sea of non-damaged DNA? We're a little confused. Is UVDDB an obligate heterodimer that we thought when we purify it, that's how we purify it? Or can DDB2 interact with DNA without DDB1? And then I'll talk about TGG. So Sam said, have audacious goals. And so this is nucleotide, this is basic excision repair, and my audacious goal is to reconstruct at the single molecule level all the enzymatic steps and be able to make an animated movie of how these proteins, what are the dynamics? How long do they sit on the DNA? Do they ever go backwards? How long do they form co-complexes? Wouldn't it be cool if we could watch that at the single molecule level and reconstitute it? But we knew doing that would be almost impossible with current technology. And so I love this quotation by Sidney Brenner. Progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. And so we had to come up with a new way to make single molecules fluorescent and study them. And so we came up with this approach called SMADNI, single molecule analysis of DNA binding proteins from nuclear extracts. Rather than purifying a protein, we said, why don't we just have the cell make it? We'll use nuclear extracts, use them directly. And we love the name. Postdocs in the lab worked really hard to come up with this name because Smadny Minch actually means thirsty monk. It's a Slovakian beer. It's not a bad beer at all. And I can always say that Matt is thirsty for knowledge, but he's also thirsty for beer. And so you can follow your favorite fluorescently tagged protein. You can watch the kinetics of it binding to DNA targets. You can do multiple colors, so you can actually watch multiple proteins talk to each other. The power of this tool is that you don't have to purify every variant. There are variants of unknown significance that we see in tumor cells, but we don't know if they're bad changes or good changes. By driving this off of plasmids, you don't have to purify the protein. We can look at post-translational modifications. And then the added bonus in the nuclear extract is that there's actually chaperones in there that can keep your proteins happy and well folded. So does it work? Well, let me show you the workflow. First, you got to get a vector, a plasmid with your protein of interest that has a fluorescent tag. I have a wonderful undergrad in the lab, Liam Leary, that grows up all our plasmids. He then gives a purified plasmid, actually send it to Plasmasaurus, make sure it's what we think it is. And then he gives the plasmid to Vera Roganskaya, who does the transient transfections, makes the nuclear extract. The nuclear extracts from about 4 million cells makes about 100 microliters of extract. And as you'll see, that's about 8 trips to this instrument called the C-trap. So what is the C-trap? Lumix has made this amazing optical platform that allows 3 very cool technologies all in one space. Laser tweezers to grab beads, this 5-chamber microfluidics that allows you to flow in different things and then observe, and then a 3-color confocal that has incredibly sensitivity. So the first step is to grab beads of 5 microns. They're coated with streptavidin. And here you can see the instrument has caught the beads in the optical traps. So think of optical traps as a laser that you could grab a basketball and move the basketball around. That's what it can actually do. And it's a very sensitive instrument. It can control the movement of the beads quite well. In the second channel, we flow in DNA. The DNA is lambda DNA usually, and it has a biotin on the ends, and the biotin sticks to the streptavidin. And then you go, well, how do you know that you caught you go fishing for DNA? How do you know you got it? Well, they've built an algorithm so that when you pull on the bead, you see a force change, and this is called a worm-like chain model. And for any length of DNA, the DNA is like a spring, and actually you can pull on it and it stretches, and that force curve tells you you caught one DNA. You then flow in your nuclear extract and watch. And so we normally do, this is called a 2D scan, the beads autofluoresce, but here's a molecule that's actually landed on lambda DNA. And then we switch to a 1D mode where we scan the DNA 10 to 30 times a second, and we image, each image is that protein. And this particular protein didn't move on the DNA, so it's a straight line over time. If it would have moved, it would be jaggy. And from this, we can get dwell times and say, the protein sat on the DNA for this many seconds. So the first question as a demonstration was, is UVDDB a heterodimer that stays together all the time? So we labeled DDB2 in red and DDB1 with EGFP, but in the instrument it looks blue. And what you're looking at is a chemograph over time. So each of these streaks are UVDDB landing on UV irradiated lambda DNA. Each streak represents an individual binding event. And what's so cool is you can see the heterogeneity in the binding events. First of all, you can see the complex landed and then left, and then DDB2 came in by itself. Here you can see both landed together, but then DDB1 went off and tried and did something else by itself because DDB2 was still there. So what kind of information can you get from these single molecule experiments? Well, if they move, you can get diffusion constants, and I'll show you that in a second. You can get dwell times, the time that it sits on the DNA, which is really a measure of the off rate. The time in between the binding events, the gap time, is the on rate. If you know the concentration, you can get a true on rate, and then you can actually get an equilibrium dissociation constant. And so he did that for UVDDB for UV photoproducts, and it was 0.4 nanomolar. So that was really cool. From single molecule data, you can get biochemical parameters. But the really cool feature is you get the dynamics, which you can't see any other way. We expected every time it would be the two proteins would land together, and that would be it. But only 32% of the time were they heterodimers. Matt wrote an algorithm, and he said, what are the chances of the different proteins coming and going on the DNA? It turns out there are 11 different types of interactions. Think that you're going to meet somebody on a blind date and have a coffee, okay? And so you're going to meet outside the coffee shop and then go in. And so if you think about that, these two met outside the coffee shop, went into the coffee shop, so they landed on the DNA together. They really hit it off, and they decided to leave together. But sometimes, here's number 9, guy showed up early, he's waiting, she arrives, they don't hit it off very well, she leaves, and then he leaves. So what I'm getting at is we can get an idea of the dynamics of who's coming and going. And this is going to allow us to look at basic scission repair 2 proteins at a time. I told you about variants of unknown significance. This is a mutation that's associated with xeroderma pigmentosum, where it's a lysine to a glutamate residue change, and it causes XP. But here you can see is now the protein, 1 amino acid change in this extract, now it's actually moving on the DNA over time, and it's blind. By taking out that 1 lysine, you can't see lesions anymore. What you're looking at is a 3-dimensional structure of 6,4 photoproduct looped out, and you can see that lysine makes important contact on that phosphate really near the damage site. So, as I said, my goal was to observe the kinetics of all the intermediate steps in BER, and so the next one I want to tell you about is PARP. poly-ADP ribose polymerase. It's an important target of cancers, specifically monotherapy in BRCA1 and BRCA2 breast cancer. In a wonderful collaboration with Brett Freudenthal and Tyler Weaver, Tyler built nucleosomes with NIEHS at 3 different places. And they have this weird superhelical location terminology. So the diet is called SHL0, SHL minus 4.5 is near the one end, and then minus 2.5 is another end. And so what you're looking at is the nucleosome, and Tyler labeled H2A with Psi3, so it's green and we can then see it. And so in a wonderful collaboration with Lumix, Marco developed these handles, which are 6 kb with unique sticky ends. And so Tyler made the nucleosomes with these complementary sticky ends, and we could ligate them together and then string them up on the C-trap. And so if we have a nucleosome, there are 2 fiducial markers on the arms that are red, and the nucleosome is green. And so you can see we caught a nucleosome, it's right there. So now we're in a position to say, does PARP work on a nucleosome with a NIC? And so this is a real data that was animated by Janet Awasa. And what you're going to see is this is a nucleosome strung up between the 2 beads. And PARP is shown here. It's YFP, but in the image it's blue. PARP is a modular protein made up of DNA binding region, 2 zinc fingers, which are going to show here, a BRCT domain, the catalytic domain, and then the WGR domain that interacts with DNA as well. So it's going to land, and when it lands, the green and becomes blue. So there's the time streak. And then when it leaves, there's the gap, and there's the dwell time. And so from these chemographs, we can get really cool parameters from PARP. And so here's an example of the work that Matt has done. We haven't published this yet, but we're very excited. The average dwell time on a NIC at 4 piconewtons is 2.5 seconds, and the apparent on rate is about 0.18 seconds. We know the concentration of the YFP labeled PARP because we can determine that by looking at the fluorescence in the flow cell, and he came up with a KD of 1.6 nanomolar to the NIC in the nucleosome. And he saw some really interesting trends, which I want to summarize here. Binding to undamaged nucleosomes increase. So PARP has affinity for nucleosomes by themselves. There doesn't have to be any damage. And when you pull on the DNA, the nucleosome begins to unwrap in a very specific way. And that was shown by Bustamani's lab. And so what we see is that the affinity increased 4-fold by just pulling on the DNA from 4 piconewtons to 10 piconewtons, which is hardly any energy at all. Binding to naked NIC DNA had the highest affinity, but then we were wrong. We thought the dyad wouldn't be recognized because we don't quite understand how PARP can actually bind to the NIC and encircle it the way it's expected to. Well, we have these NIC nucleosomes. The next enzyme in the pathway is to look at ligase 3 and XRCC1. And here, a wonderful postdoc in the lab, Ashna, has labeled ligase 3 with halotag 635 red and then the YFP labeled, which looks blue here. We thought these proteins, and you purify them as obligate heterodimers. But what you can see here, and it's really quite apparent, they landed together, but then XRCC1 left and just left ligase 3. And then XRCC1 came back, and then ligase left. So this is amazing. We can actually watch the kinetics of these comings and goings of proteins. So they bind together, XRCC1 dissociates, XRCC1 reassociates, and then ligase disappears. So she's found that non-damaged nucleosomes are bound by ligase and XRCC1. So this is really amazing. This says they have affinity for the nucleosome. So if we're going to get there, we need to look at a lot of proteins. And I just recently gave a talk over in Amsterdam at Lumix, and I said, guys, let's put a table together of all the proteins you've looked at over the last 18 months with dwell times. And I was stunned. In 18 months, they've looked at 30 proteins or protein variants using the SMADNI approach. So this is just incredible. I know we're running out of time, and I just want to talk about this really interesting glycosylase involved in oxidative demethylation of 5-methyl-C, thymine DNA glycosylase. Whenever we go down to the C-trap and we see something new, I like to get a picture of the team that was doing it. So this was a really important day. We went down there with the extracts, and right off the bat, Brittany was able to see TDG. And you can't see it, but they're smiling ear to ear. So I told you that thymine DNA glycosylase is involved in oxidative demethylation. 5-methyl-C gets oxidized 3 times by this protein called TET. And the last 2 oxidation steps, formal C and carboxyl C, is then removed by base excision repair through TDG. And in a wonderful collaboration with Alex Strohat, Brittany's first question was, can we see, does UVDDB stimulate the turnover of TDG? And so this is looking at the excision of formal C, and you can see that the enzyme by itself doesn't do so well, but when you add UVDDB, you get this 14-fold increase in the catalytic rate. So UVDDB can help TDG turn over. Well, how does TDG behave on DNA? And that's what I want to show you. If you're a protein looking for a lesion, you have some choices. If you're high enough concentration, you can be like a helicopter, land on the DNA and say, oh, is there damage here? No, okay, I'm going to leave. That's just 3-dimensional diffusion, not very efficient. Another way is, well, I'm going to land on the DNA and slide. Well, unfortunately, in a nucleus, you're going to run into another protein like a transcription factor or a nucleosome. So some proteins can actually jump or hop along the DNA. And so we wanted to investigate, how does TDG find lesions? So we took lambda DNA and nicked it 10 times, and we incorporated using nick translation a formal CTP and also a fiducial marker, DUTP. So we knew where to look for the formal C lesions. And so we could say, does TDG land on the damage or somewhere else? And so this is one of the first chemographs that Brittany showed. It's beautiful. You can see the blue streaks. That's where the damage was, and it photo bleaches. And you can see 2 types of binding events: proteins that land non-specifically and slide or hop, and then these long-lived guys that sit on the DNA for a long, long time. You can analyze these data. Oh, this is a wonderful, if you were actually able to watch the single molecule, this is what it would be looking like as it's moving on the DNA. So we analyzed by, it's called a cumulative residence time distribution, and the non-specific are very short-lived, and the specific are very long-lived. Oh, what's it? Sorry. Going the wrong way. Okay. I'm almost finished. So this is really interesting. It looks like 2 proteins actually came together, and we're like, did one hop over the other one? I don't know, I can't tell. Well, using extracts and halo tags, we could actually make green proteins and red proteins and mix them together, and you can see that they actually jump over each other. So this is proof that they hop. We did a salt experiment, and if hopping is occurring as you increase salt, the diffusion constant goes up, and that's what we saw. This is the coolest experiment, and I'm going to leave you with this. If it can hop, it should be able to hop over a nucleosome. So we happen to have nucleosomes in the lab, and what you're looking at is amazing. Here's the nucleosome, and TDG slides in, crosses, hops over the nucleosome, slides back, but now it's interacting with the nucleosome. It's sitting on the nucleosome. Nucleosome has no damage. This is the first evidence that TDG actually has affinity for nucleosomes. So that's pretty cool. Brittany came up with this idea that the unfolded 82 amino acids of TDG might be important for binding to the nucleosome. So when we deleted that, it no longer interacts with the nucleosome. It's shown here. So this is really cool. So stay tuned. So I just want to say that the advantage of SMADNI, we can look at variants very easily, protein post-translational modifications, chaperone stabilization, increased specificity. We think there are dark proteins that actually increase specificity and also help with facilitated association. And with that, I'm going to close and acknowledge an amazing team of scientists in my lab over the years. Some of the work that I've talked about today is highlighted in blue. As you know, the Hillman Cancer Center has an amazing genome stability program. We like to call ourselves the DNA pit crew. And I co-lead the program with Patty Opresco. Amazing collaborators, including Sam, Tremendous support from the Lumix team. And finally, I want to end by thanking NIEHS for this amazing grant that I have. It's called the R35 a River Grant. And I have probably the best program administrator you would ever hope to have. Michelle Heacock, she's always excited about what we're doing. Send me slides, let me see. And I really appreciate it. So thank you, NIEHS. And with that, I'll close, try to answer any of your questions. Thank you very much.
So we have time for a few questions, but before we do that, two things. I want to acknowledge the Wilson family. Dorothea Wilson, Joe Wilson, and family are here today to see this event. And thank you, Ben, for accepting the invitation. So I also want to mention, after this questions and everything, we invite the Wilson family, Ben, and friends and colleagues of Sam Wilson to join us. We're going to take a little journey out to the Memorial Garden, where we're going to dedicate a special bench that Bob Petrovich helped put together in honor of Sam's legacy. So we can take questions up here, and then there's probably several Zoom questions. I'll start.
Thanks, Bill.
With a question. So the last one, the TDG, when it binds DNA, it's going to help recruit other proteins then. Does that recruitment require the presence of DNA, or can you see this interaction with other, you had that interactome TDG. Do you think, does that interactome require DNA for interaction?
Oh, so that's great. So Matt Shy, a postdoc in the lab for his K99, wants to look at, does it interact with TET? So he's working on that grant proposal right now. We've started some preliminary work with making extracts with TET and TDG, and we haven't looked at that. You know, and CPG islands are associated with heterochromatin. And so one of the fundamental questions is, how did they get into the heterochromatin, compact chromatin, to do what they do? And so are there chromatin remodelers? Is UVDDB involved? And so we're in a great position to go after that. Well, hopefully Matt will go after that for his K99R00.
Good.
Thanks for the question.
I think Michelle's got a question, and I'm going to see if there's Zoom questions.
Yeah, I wanted to say, Michelle, that I specifically started the nucleosome work because of your pestering, so I appreciate it.
Pestering?
No, are you going to do nucleosomes?
All right, so I've got lots of questions for you, but I get to meet with you later. But maybe just your last story that you told us about TDG, it jumping off nucleosomes. Can you tell us a little bit more about how it manages to stay in the vicinity and is able to hop back on?
Yeah. So one of the problems that we have is we can't appreciate, once a protein goes into solution, how fast they're moving. So what's happening, and the way I think about it, it's like it's a hovercraft. It leaves the DNA, but there are forces that bring it back to the DNA. And so it basically isn't going macro dissociation. It's just micro hopping and can actually just move a little bit. And it's going in all directions, and the likelihood of coming back in the same direction to hit the DNA, it happens. And so a lot of times it'll just hop over things. Thanks for the question. I'm looking forward to speaking with you. Tom.
Ben, do you have examples where you introduce a change in a protein and change its single molecule behavior that correlate with an effect on base excision repair in a more biological context?
Yes. OGG1, we've made the catalytically dead OGG1. It can bind to 8-oxoguanine, but can't break the glycosidic bond. And that protein sits on the DNA much, much longer. So it's basically sitting on 8-ox, but it can't remove it. So it sits there much longer. Didn't have time to show that data. We've made several mutants of TDG. We've made the equivalent catalytically dead TDG. And we had a bet. I lost the bet. It doesn't bind near as long as the wild type. So something about that particular residue that's important for breaking that glycosidic bond is also important for binding to DNA. And so I think every glycosylase we're going to look at is going to be slightly different. MPG we've begun to look at, and too soon to say for sure what's happening there. Thanks for the question.
We've got a question online from Nathan. I don't know if you can let Brett Frudenthal, as you know, was Wilson postdoc and now is at there it is, Kansas, University of Kansas. It's good to see you, Brett. Thank you for joining. If you,
Yeah. Hopefully you can hear me. Sorry, I didn't know I was going to be unmuted, but yeah, great talk, Ben. Sam would be proud. I was curious on your TDG stuff. It looked like, I know it's just one chemograph, but it looked like maybe the N-terminal domain maybe is involved in mediating some of the interactions between TDG and the DNA. Do you think it is as well? Like, just out of curiosity, or can you comment on that?
Yeah, so the intrinsically disordered N-terminus, that was Brittany's original idea, that it was important for its diffusion. And so before the mutant was made, she discovered that it binds to nucleosomes with your tremendous help. And so then when the mutant came in, we could actually answer both of the questions. Is it important for binding to DNA? And the dwell time is shorter. You had a good eye for that chemograph. And yes, it seems like it can't see nucleosomes anymore. Thanks.
Thank you.
Hi, Joel.
Hey, Ben, awesome. So you showed the data of what happens with UV DDB loss or knockdown or decreased amounts. What happens if it goes in the other direction? Can you overexpress? If you do, are there downsides with that, or are there upsides with that?
Yeah, it's a really important question, and it's a technical detail that many people worry about. All the experiments that I showed you with IF were driven off of plasmids under transient transfections. So by definition, the protein is overexpressed. Now with CRISPR technology, we could go in and knock in the label and ask under physiological conditions, what do we see? And one of the leaders in the field of nucleotide excision repair that's doing that is Wim Vermeulen for all the nucleotide excision repair. And he definitely says, if you overexpress the protein, the cell's not happy, and repair doesn't occur the way it should. And so thanks for that question. And with CRISPR technology, we could do that. We have a stable cell line that expresses DDB2 M-neon at 3 times over the endogenous level. And that protein works very well. And we actually use that to look at its interaction with OG. So I think that interaction is absolutely real. But thanks for the question.
so Ben, I'm just going to read some questions for the sake of time. Jan Liu at the University of Florida International, also from Sam's lab, has a question that, do you think TDG interacts with modified histones?
That's a great question. And Epi's cipher sells modified histones. And so that will be part of.
There's John.
Yeah.
Hi, Ben. Thank you for the amazing talk. It's very exciting. Congratulations for all the findings. I'm just for curiosity, if you think about like modification, is that possible the TDG will be interacting with some histone modification to help the hopping?
The next experiment that she's going to do is just express those 82 amino acids with the halo tag and say, is it necessary and sufficient to bind to nucleosomes? If that occurs, then we can start testing what are the interactions and do modified histones affect its interactions? Great question.
Well, cool. Thank you.
Thank you.
So Trevor Archer had a similar question. I think you just addressed that. Other than that, we had some congratulations online from Phil Hannawall.
Ah, wonderful.
Olga Laverick.
Oh, wow.
And Setra Narayan. And I think that, and me, and Rob Sobel.
Ah.
And they're coming in. So it was widely attended virtually as well.
Oh, thank you.
So I think with that, I want to give Sam another round of applause. And please join us to a little tour in the garden.